this is the greater UCL. Right, these are all the different departments. It's a multi-faculty university, so it's not just like Imperial, which is just really the medical sciences. It's got many other standard university faculties, basically. It has many different locations. I don't know, do you all come from London, or are you all from
all over?
All over the place. Okay, so basically you have the main campus of UCL, which is near Euston, off Gower Street. So as you come out of Gower Street, Bloomsbury, that's the main campus where most of these faculties are, and quite a few of these as well. You have the Royal Free Campus, which is this one here, which is based on the hospital site. So here's the old medical school, and the hospital's there, goes up to 12 floors. Institute Child Health, which is where Great Ormond Street is. So that's the institute which goes along with Great Ormond Street, the Children's Hospital. Then you have the Institute of Ophthalmology, which is the eye, associated with the eye hospital, Moorfields, which is in the city, just off Old Street. Institute of Neurology, of course, which is at Queen Square and is associated with the National Hospital. And then you have the Whittington Hospital at Archway, which is just along from here in Highgate. So they're the main sites. There are other smaller sites as well, but they're the main sites, and these are the hospitals which are associated with those. And this is the so-called medical school, or School of Life and Medical Sciences, which encompasses 4 faculties. And the brain sciences is where we are. So this is us, Department of Clinical Neurosciences, and we are in brain sciences as part of the Institute of Neurology, but at this campus. So we're fully integrated within Institute of Neurology and UCL, and in that part of the university. So we can also come to the medical schools.
Brain sciences is a faculty, is it?
It's a faculty, yes. So this has all been revamped for probably the 4th time since we merged. But I think this is probably the one which is going to last.
The next one.
For the rest.
Yeah, so it's quite big. It's one of the bigger faculties, and it encompasses a lot of different departments within that. So there are departments within Institute of Child Health, obviously a lot with Institute of Neurology, but also lots of the main site of UCL. So UCL has the biggest neurosciences research in the country, and it's one of the top in the world for neurosciences. That encompasses not only the clinical neurosciences associated with the disease itself, but also way back to the basic neurosciences. So we regularly have UCL Neuroscience Day down on the main campus, when there are, I think, over 500 people attend that just from UCL, and have a full day of neurosciences. So there's a lot of neuroscience goes on at UCL at various different degrees in various sites, and we're just part of that, obviously. Okay. So within the department, we share the facilities between everybody in the department, and this is the type of research that we do. You may have heard of some of these: Friedreich ataxia, mitochondrial disorders, Huntington's, motor neuron disease are probably the main ones, and peripheral nerve disorders used to be a major one with Professor P.K. Thomas, and he ran his group, was again a world leader. But probably the bigger focus of the department in recent years is devoted to Parkinson's disease. I'd say at least 3/4 of the research that goes on in the department is now related to Parkinson's disease. And there is a relationship also with mitochondrial disorders, how they overlap as well. And
has this always been acknowledged, that link, or is it kind of a research? Because like you hear quite a lot recently, the kind of that aspect of Parkinson's, that it's mitochondrial problem.
So my history is I did my PhD many years ago. I got my PhD in '87. My PhD was on mitochondrial disorders, which wasn't related to Parkinson's at the time. And at the same time I did my PhD, I don't know if you know the MPTP story, which was the drug addicts taking MPTP accidentally developed Parkinson's disease. That came out at the same time I was doing my PhD. We then looked at mitochondrial function in Parkinson's disease then, and we were the first people to produce a paper demonstrating mitochondrial dysfunction in Parkinson's disease in 1989.
Right.
And that's been cited over 1,000 times. It's very highly cited papers. Then So there's a lot of excitement at that time, and then it quieted down a bit because people didn't know what to do next. And then when other things come through, there's now a thread of research as you look through since other gene mutations have come through, just the mitochondrial involvement, probably 8 or 9 years ago. Again, it's built up again, showing that mitochondrial dysfunction is likely to be playing a role. Exactly where that role is, whether it's causing the disease or a consequence contributing to the disease, is still debatable. But yes, mitochondrial.
Is it correct? Have I heard right that that there is a drug that will stop the decline of the person? This halts it. Doesn't cure you, but it's on trial at the moment.
It's the Rasagiline. I think it came.
That affects, that targets mitochondria, or just generally, you mean? Do you mean?
Well, it stops the decline
Of the disease?
of the disease.
Is the Rasagiline that the paper come like 2
Okay.
months ago? So they show that when it's novel patients just after the diagnosis, and what they did is give this medication or give levodopa only, I think, as comparison. And what they show is that they followed 2 or 3 years, and so that it was the comparison, it was less decline in the motor symptoms. So it's not so clear yet that can neuroprotect, but what it looks is if you start early, at least can delay. But I think they are doing more studies now.
So we've got to hang in there a bit longer.
So it looks as if it's slowing down rather than stopping.
Yeah, but I think stopping, it would be quite tricky. If you at least get slow a lot, it could be good enough, or well, not good enough for you, obviously, but in general, I think it could be a big improvement, because now is nothing working really in terms of decrease the progression, slow the progression.
So where's that in terms of clinical trials?
So I mean, as far as I know, Rasagiline have been used for a long time. It's a lot of papers, but it was recently, some years ago. It was the Adagio study. It was with Fabrizio and other people study. So they start to compare patients who start only with levodopa or with this, and they thought, it's an improvement. They have less decline. So then I think it's when they really start with the first clinical trial, design multicenter to compare, because a lot of times they know what the patients have. So it's more difficult. So I think it was around 2006, 2007 when they really start with the clinical trials.
Yeah.
So,
we went to Imperial College last week, and Professor Abramov was talking about this as well. That he said that Rasagiline turned out not to only, because it was originally designed to influence the reuptake and that kind of flatten out the availability of dopamine, but somehow as a secondary side effect, so not necessarily side effect but effect, it was actually reducing oxidative stress and
my doctor is Professor Roger Parker.
Oh yes, in
Cambridge.
Cambridge.
And he's dead keen on this stem cell.
Okay.
Drilling holes in people's heads and planting stem cells.
They started that again, haven't they?
No, they start already. Told me the other day,
Because a lot of that was
was
done 20 years ago,
Yeah,
and I think they're revisiting it again with improved methodologies.
Yes.
I've heard at some lectures that that's a complete waste of time. You wouldn't know where to start with. Where would you put the stem cell to start with?
Well, I mean, again, it's not an area we work with, obviously, so it's not something we can directly comment on. But yeah, there are issues in terms of the type of treatments that you can look at are identifying the disease early or pre-symptomatically and try to get what's known as neuroprotective therapies in, so that you can slow down or prevent the disease progression before you get the disease, so predict who is going to get the disease and then stop that happening. The other one is to try to slow it down or prevent it progressing once you've got the symptoms. And the third one is, once you've got the symptoms, to actually improve the symptoms you've got, is to replace the cells. And to replace the cells, either you need the cells to replicate themselves, which is more difficult because brain cells don't like doing that, or you have to inject cell replacements. So there are different avenues. of where you come from, and there are a lot of problems with the brain which you may have come across before in terms of getting the therapy to the brain. I'm sure Lydia will talk to you about that a little more with her project.
What ethics? Are you talking about ethics?
No, not only ethics, but also getting, so when you're doing treatments, if you want to get things into the brain, lots of chemicals or substances do not go from the blood into the brain. You have to directly inject into the brain, although Lydia will tell you that's not necessarily the case. So we are coming from looking at what may be causing the disease as a general ethos of the department, people working within the department. So what's the cause, and then to understand how those causes lead to the disease, what's the mechanism. So if you understand the causes, this also helps you understand the mechanism, and then you can identify how you can intervene. What do you have to start changing or modifying, or what markers can you see here, so you can see people who have not yet developed the disease, but you'll be able to predict who will develop the disease before they actually get the symptoms, and then if you can put protective therapy in at that stage, then you don't need the cell replacement. You can stop the cells dying in the first place. So that's a lot of area where we're looking at. And to do, I mean, there's obviously many things we're doing. I just want to give you a brief overview because you obviously.
Should we let you sort of do your talk then?
No, I'm happy to talk in, but obviously I don't want to take away from anything. But I'll give you an overview, and I'm happy to discuss anything else and anything Lydia may be talking about as well. But so one way we look at this is to generate models of the disease, so we can understand some of the causes and then try and model that, and then study how those models cause the disease, which are also useful for when you looking for therapies. You can use those models to test your therapies before you go to patients. So to get the approvals of the authorities to give novel therapies to patients, often, in most cases, you need to demonstrate that A, it's not deleterious, and B, can be effective in a decent model of the disease. And then you go on to use it in early phase trials with patients, and then further on. So some of the models we generate will hopefully add to that understanding as well. So if you look at this is Parkinson's disease in general, all the patients with Parkinson's disease. There's probably, it's debatable who you speak to, but there are about 10% of patients who will have a known genetic cause. That's increasing as we're speaking, but it's still small. It's, 10%, 15%, maybe 20% in some people's eyes, depending on some of the screening which is being done, how you interpret that.
So it's 10% of the people with Parkinson's?
Yes, that have a genetic cause.
Yeah.
And some of the main genes which are involved are these ones. There are many others, the whole list is growing and growing and growing. But alpha-synuclein gene, LRRK2 gene, GBA gene, glucocerebrosidase, Parkin or PINK are probably the most recognised ones, but you can have a long list now.
I've been sort of testing this familial link in various meetings, and on average it comes out at about 30 to 40%.
Total.
People who have Parkinson's who have a known relative who also have.
Right, yeah.
And that's second degree relatives. It's about 25% when it's first degree relatives.
Yeah.
And I suspect that almost seems to be the area between the two dotted lines there.
Well, also if you then take a certain population, so there's a population in North Africa, the Berber population or the Ashkenazi Jewish population, where up to 40% of that population has mutations of these two genes, right. Either or. Yeah. But in the UK, it's around about 3% will have mutations of this gene, up to 10% will have mutation of this gene in the UK. So they make up most of this 10%. But like I say, with all the modern genetic analysis, which we collaborate with John Hardy, Nick Wood up at Queen's Square, as well as throughout the world, they're coming up with other places in the genetic material which is a higher risk of Parkinson's disease, and actually coming up to exact numbers of which genes and how many patients are involved will be resolved in the next 5 or 10 years probably, or maybe slightly less. But if you look at all the other genes, and many more, which come as linked to the disease, there are many of those, and more of those mutations will come out as they do more detailed sequencing of the DNA. So what we're interested in is studying these mutations, trying to understand why these lead to cell death and disease, and how that may help us understand what may be causing the other patients. So these are the sporadic cases where there's no known genetic mutation yet, whether or not the environment plays a role, so in other words being exposed to certain environmental aspects, toxins and things like that, whether it's a combination of genetic and environment, or whether or not there are other factors that can also influence what's known as epigenetics, which is not the genetic mutation itself, but how your environment affects your genes itself. So you've got different contributions there. So there are two types of model we use, and Lydia will refer to these in her talk as well, I think. So one is where we take cells in culture and model a disease in those cells. And the other one is where we take mice models, and get those mice to express the mutant proteins as well, mutant genes, mutant proteins, to try to see if we can get a model to work with. Although most of what we do is actually cell-based, because it's more amenable to our type of research. So just a quick guide, hopefully if you've got any questions on this, please ask. But so a quick guide, what you have is you have a cell here, and here's a diagram of the cells we use. These are similar to brain cells, neuronal cells, and what you can do is you can insert what's known as a vector, which expresses the mutant protein or mutant gene which causes the disease in certain people. You get this cell then to produce the same mutant gene, mutant protein, and that then we use that to then model the disease. So this is producing the same mutant protein that the cells do in people with Parkinson's disease with any given mutation which we're dealing with.
Can you actually perform that on a one particular cell basis, or it's always like a big collection of cells?
No, you can do single cell work. So you spoke about Andrei Abramov, who does single cell work. We do the same type of single cell work as they do as well, in different ways. So you can analyse them, see what's going on. So here you have cells, not the same cells, but this green is where we can stain. So some cells into this which have been injected with this vector to produce this protein, we can then stain that protein using antibodies to specifically see it's expressing that protein. And then you can do various analyses of that. It could be whole cells, many millions of cells together you analyse, or it can be single cell analysis where you look at single cells. And you can then go on and develop that into more neuronal type of cells. So these are ones which are showing more neuronal features similar to what you'd see in Parkinson's disease. And we analyse all these type of mutations. My focus is predominantly on alpha-synuclein because that's linked to the disease very clearly. Because this is a protein which aggregates in the brains of everybody that has Parkinson's disease. So we study this a lot. That's something Lynn will be talking about in more detail. And likewise in the mice, so here we can again get mice to express the same type of proteins. So you put this new gene into the mouse's DNA, and they then express that mutant gene, which gives you a mutant protein in various places in the brain. So this is a slice through the brain of a mouse. So if you take the mouse brain and slice it down there and stain it, this is the mouse. So here's what's known as the cerebellum. That's the back of the brain, and here's the front of the brain with the cortex, and the area affected in Parkinson's disease is around here. And so that's a control mouse, and this is stained for the mutant protein which accumulates in people with Parkinson's disease. So this accumulates, and you can see it dark staining. And if you look at a high magnification, this brown dotting is where the protein is aggregating.
I was assuming the mouse hasn't got Parkinson's anyway.
No, this mouse it does get mild movement disorder, very mild movement disorder, but not very few of the mouse models give you Parkinson's disease, whichever model you're dealing with.
Right.
Very few. Some of the chemical models do, where you give them a toxin, they are more similar in some ways. But where you're trying to model the genetic causes of the disease, where you put the gene into the mouse, very few show you typical Parkinson's disease in a mouse, but they do show you some features. So this particular mouse we're dealing with here has some clinical features when they're around about 18 months old. And the main thing here we're looking at is the fact that they produce the same inclusions as you see in Parkinson's disease patients in the cells of the brain. So this model is very good if you want to address the protein aggregation. And Lydia, again, the reason I've introduced this to you is because Lydia was dealing with this one. Okay, so hopefully I've tried to give you an idea of what we do, a brief introduction. Again, quite happy to talk anything about that now, or if you want to let Lydia talk about what she's doing.